HIV, it's a very known virus in our days, and HIV is the virus that is responsible for one very important and serious disease, which is known as the AIDS. In this video, we're going to talk about replication of HIV. We're going to see how HIV is able to survive and replicate within humans. But something that I want to make clear here is that HIV is the virus and AIDS is the disease caused by HIV. Now, AIDS, as you all know, it's a very serious disease, and it's a disease that caused more than 20 million deaths so far in the whole planet. Now, the main way that HIV is transmitted from a human to human, it's usually by sexual intercourse. Of course, there are other methods, but the main way of transmission is sexual intercourse. The good news here is that HIV is not airborne. So imagine our problem if HIV was airborne and was capable of transmitting through air. That means that many people will have been infected with HIV so far. Now, HIV, it's a retrovirus. Now, what are retroviruses? Retroviruses are simply viruses that do not have DNA. They have RNA as their genetic material. And let me draw here, let me take another color and let me draw the virus. So imagine this is a virus here. Retroviruses, they usually have RNA. So their genetic material is RNA, not DNA. And when you have RNA, it means that you need to have some enzymes to go with it. So I will draw some enzymes here. And these enzymes, we're going to see one specific enzyme later on and why it's important for the replication of HIV. So what you need to know here is that retroviruses are basically viruses that contain RNA with enzymes. So the genetic material and the enzymes are very well protected within the HIV particle. Now let's see how exactly the HIV replicates in humans. So let me draw here a big cell. So imagine that this is a human cell and it contains all the typical organelles. So I'm going to use the red color to make the nucleus. And inside the nucleus, obviously, you are going to find the DNA. And of course, here is a cytoplasm where you are going to find all the other organelles, like mitochondria or the Golgi, the ribosomes, and all the necessary organelles for survival. Now, HIV cannot bind to any cell in the human body. So that's the HIV here again. And HIV can attach only to specific cells, and it usually likes to attach to immune cells. Some examples of cells that the HIV likes to bind are the macrophages, which are responsible for doing phagocytosis, and of course the T lymphocytes. These cells are cells that are responsible for starting and coordinating your immune response. So especially because there are many types of T lymphocytes, the one type that is very much affected by HIV are the T helper cells. But these are cells that we're going to talk in another video. This is just to give you the idea of some cells that the HIV likes to bind and attach. So the first part of replication of HIV, it's when the HIV is able to attach onto a cell. So here is HIV with its RNA and the enzymes that are needed. So the first stage in HIV replication is the attachment. Upon attachment and upon recognition of these two cells, the contents of the HIV are going to be released in the cell. So the RNA here, together with the enzymes, are going to enter the cell. So this is step 2. Now, step 3, it's something amazing. It's something that we haven't seen before. When HIV was first discovered, we didn't really know that viruses can actually do this kind of mechanism. And what mechanism is this? We learned in previous lessons that DNA, during DNA expression or during gene expression, the DNA is transcribed into RNA. Now, the HIV has one enzyme that is called reverse transcriptase. So one of these enzymes that I talked about, let me change the color again. So one of these enzymes that I talked about before is known as reverse transcriptase. And by the name, you can understand that this enzyme will do reverse transcription. So what is reverse transcription? Basically, instead of going from DNA to RNA, which is the normal transcription, we're going to go from RNA back to DNA. So that is reverse transcription. And what that means for the virus? It means that the virus, it's going to convert the RNA that it has into DNA. So from one strand, the genetic material will become two strands. Now the DNA that is created, it's going to use other enzymes in order to insert itself into the nucleus of the host cell. And again, the host cell, it might be a macrophage or a T lymphocyte. So step 4 will be when the DNA of the virus, or the viral DNA as we call it, will get incorporated, will get integrated with the human or the host DNA. So here we're not going to have a mix of the human DNA with the viral DNA. The insertion of the viral DNA is completely random. We do not have a specific location for the viral DNA to integrate with our DNA. It's completely random. Now what happens afterwards? Now in step 5, so here step 5, it's basically when humans now, because of course we're not aware that we do have a viral DNA. Sometimes we can recognize that we have a viral DNA, but some other times we cannot. We are going to express the information that is inside this viral DNA. And as you know, the DNA contains information of how to create something. In this case, the viral DNA contains information of how to create the virus. So we are going to express these genes, and we are going to start synthesizing more viral particles. So here we're going to create more HIV particles with their RNA and their enzymes, their necessary enzymes for their replication. And then when we make a lot of them, eventually these viral particles will start escaping. They will start exiting the cells and entering the bloodstream. In that way, these viral particles here, these HIVs, will be capable of infecting even more cells and replicating even more. So this step here for 4 to 5, it's also very crucial because the virus will take advantage of us. It's going to use our machinery, our tools, our enzymes, our ribosomes in order to express its genes, make more viral particles which they're going to spread through the bloodstream into more and more cells. So that is the last step of HIV replication cycle.